So welcome to the reading group. Matt, you've shared a delightful overview of these different practices for cultivating heirloom microbes in lots of different corners of the earth, from Mongolia to the Alps and elsewhere. And you've also brought our attention to some really important work on phage. And the piece that you shared with us on phage is actually something we're going to talk again about in a few weeks with the author, or the senior author on the paper, whose first name is Forrest. There's a moment in your paper where you invoke phage, but maybe before we get to phage, I just had a broad question about, Heather Paxson has talked about modes of sort of sustaining these microbial cultures in a post-Pasteurian landscape. And thinking about these very different sites that you've brought us to, could you talk about the different strategies, not just of, this is obviously all in places where it's moving beyond a Pasteurian logic, or maybe not even approaching a Pasteurian logic. But what are the strategies for, you've described them ethnographically, I think, but do you see any strategies that kind of travel, or are they all particular as people approach these different fermentations?
Well, I think there is definitely a lot of traveling norms in that sense, that this European or Euro-American idea that swift cooling is extremely important for milk hygiene. This is definitely something that a lot of people also outside of Europe and America would subscribe to. But of course, there is, as I think I'm trying to show, also in Europe, there is a lot of people who are critical of this practice, and all the way to microbiologists. Like there is a group in France who has been working on this issue for 20 years, and they clearly they make also the scientific claim that cooling milk is actually not very good for cultivating it, for keeping the already prevalent microbial community alive and well. So there's definitely a lot of different countercurrents that are happening, but I think and as important I think it is to support forms of cheese making that David Asher calls natural cheese making. I guess it's but and maybe a post-Pasteurian way of dealing with the microbial abundances that we find in milk. It is, I think, also important to say that this is a very minoritarian position. So even among the artisanal cheesemakers that Heather Paxson works with or used to work with in New England, I think there's very few who actually go all the way to make these natural waste starter cultures, because they are just a lot of work, and of course also a riskier form of culture care than if you buy your clarified, clear strains that basically where you know what you get, and especially when you work with raw milk. And it is, of course, a lot of as many agricultural products, there is very small margins. So you really have to be able to afford also a level of or an amount of bad productions that, of course, is so that's difficult. So there, but then of course, when we look at Mongolia, of course, there is a very different form of thinking these things through. There is no refrigeration on the Mongolian steppe, or people might have a small refrigerator hooked up to a solar panel, but of course that's not nearly big enough to really cool the milk production. So in that sense, I never had the feeling that the herders in Mongolia had this idea that cooling milk would be a good idea. But what they do, and we found that interesting, that we couldn't get to the historically so far, we couldn't get to the bottom of it, that they almost all the milk is boiled right after milking and then stored. So they basically, they do a form of pasteurization that for a raw milk cheesemaker kind of breaks my heart. But this, of course, is a very different form of making cheese, and also kind of looking at the whole process that is kind of goes through a different biochemical process than European style cheeses. It makes a lot of sense actually to boil the milk in advance and then kind of keep it together for 2 or 3 days.
Thanks. And others, again, feel free to just unmute yourself and ask questions or type questions in the chat. But I've got another quick follow-up, just sort of onto epistemological one. So you talk about how the companies that are involved in this space have tried to, hoard the knowledge and proprietary databases, and a little bit about some strategies for resisting that. But could you just walk us through? So there's the one story where the cultures are purified and then sequenced. So I guess the rationale behind that purification process is to just identify one microbial genome at a time. Is that kind of the logic?
You're talking about the dairy culture companies, or so not about the genetic research that I'm part of?
There was the one like close ethnographic story where you're following somebody around the lab. They're having to do multiple things in real time to respond to the microbes, and they're culturing the bacteria and then sequencing them.
Right.
Is that just to isolate the strains before?
Yeah, so kind of that also took me quite a few attempts at kind of, or a few tries, and kind of asking my colleagues why it is so complicated. So we've been collecting dairy samples in the Alps, in Jordan and Mongolia for the last 3 years, and so the idea was to get the raw milk, but then also get as many different kind of time stamps in the process. So let's say we get the raw milk, we get the skimmed milk, we get maybe the cultured milk or the pasteurized, or kind of, or the boiled milk, to then see, also in an attempt to hopefully be able at some point to get a kind of a time way diagram of microbial community through a raw milk or kind of, or peasant fermentation process. So these, of course, are always communities of microbes that are in there, and in order to get those into the genetic sequencing pipelines that have been built at our institute, we actually send them to a microbiology lab to do more classical culturing work to amplify these. So kind of, as you said, kind of to culture them, isolate specific strains, and then grow more of this strain, so that there is just the quantity is. So if you just take the kind of the microbial community and kind of send it through the sequencing pipeline, it would be extremely difficult to get to strain specific differences through these, because just the amount would be too small. So that's why we kind of need this amplification step before we then kind of feed the so purified single strain cultures into the sequencer.
Super interesting.
But even that is, so I think as I'm trying to show in the article, it's been a really interesting but bumpy ride, and very and I guess also as someone this has been my first project, like working with scientists, and I also kind of from time, like at several points, I felt so naive that I just thought, okay, I mean, they just have their machines, why can't they just put them in there and then they get a result, and that's how they make science. To see that probably it's true for most of science, but I guess this project is even more challenging because it's so because nobody has really done that work. And when you look, of course, there's as I also show, there's been a lot of work on in the dairy industry to identify these good strains that you can use for these commercial starters that often are 2 to 4 or 5 different strains that are mixed in some way. But if you really want to get to the abundance and to the community level of milk, it's nobody has really done that work, and of course it's also for many reasons they haven't done it, because it is very challenging and it is extremely expensive to collect these samples, to build up a cold chain, and of course there's also a lot of hope in with my colleagues with new forms of sequencing technology that might actually for the first time make it possible to really sequence in the field, so that this of course would open a whole new world of data production.
Mm-hmm. Mm-hmm. Is that good?
Yes, I have a direct question. Matthäus, it is amazing. I mean, your piece, you nicely weave all the stories together. It's going to be one of my model articles to write in the future. I'm just wondering what you just said. Do you see a risk to reduction of all the complex interaction to the only strains and genetic numerizations? I mean, I feel that this kind of, when I read this kind of works about the genetic sequencing of the microbes and other things, which I feel that have the risk to reduce all the complexities we have in the fermentation practices, just a list of the strains. And yeah, does it make sense?
Yeah, I think, yeah, totally. I think there's definitely a danger of this happening. I think at the same time, for people who are actually working with milk, I'm not so sure this makes too much of a difference. So I found it always interesting, and of course this is also kind of in a way a big problem now that 3 years after we started, we still can't give the people who gave us their milk clear answers, because they were just extremely curious. They want to know, and of course by now most of them are they have an understanding of what bacteria are, and so they're just curious, what's in my milk. But I never felt like this would be a kind of, that this would change their interaction with the milk in a substantial way if they just get a list of the microbes that are in there. All of them are just very attuned to, or kind of they know that kind of how different it is from day to day. I mean, not every day, but there's of course, like when you work with raw milk, it's just a very lively substance, so it changes, and a lot of these changes. And I guess even if you had like a whatever, like high-tech lab in your dairy that could tell you in 5 minutes what's the milk today, I don't think that a lot of people would actually use that knowledge in kind of for their practical work. So there is some people in the raw milk sector who have kind of a very clear thing about tasting the milk and then doing different things with it. So I think there's many ways of sensing milk that are, I think, yeah, I would still say superior for a cheesemaker than kind of the knowledge of the particular species in there. And of course, one thing that might take a long time to really get to the bottom is strain-specific qualities. So there is so many different. We know that kind of, and Evan has done great work on this, that the species category is just not very helpful when we look at microbial communities and what they can do and what they can't, and especially when it gets to like producing flavors, which is such a complex thing that I don't think microbiology has a clear understanding of how it actually happens. So it would have to go down to like really minuscule gene expressions that I don't think that this is something that. So I mean, not to say that this might be happening at some point, that people kind of, and I'm sure there will be these these promises and these hopes that you could actually engineer your cheese by engineering the microbes that you put in there. So that I think is, again, always simultaneous processes that are happening side by side, even if they don't interact too much.
Maybe if you could give a little more context about the multidisciplinary team that you work within, and in that context, could you talk about what the Heirloom microbe project might look like in a future-oriented sense? You know, we've got various kinds of seed banks for plants, and now, you know, frozen arcs for endangered mammals and things like this. In the context of the broader biobanking movement, what does the Heirloom microbe project aim to do?
Right. So yeah, that is, and I think it also is one of the reasons why the I mean, the project is so convoluted, and I think also the article got a bit I mean, yeah, convoluted. It is, so the idea was by two archaeologists and archaeogeneticists, Tina Warriner and Jesse Handy, to look at the early spread of dairying across Eurasia through deep genetic sequencing of microbes. So I mean, there's the archaeogenetics component, but there's also a proteomics approach, because milk proteins are some of the most stable organic small particles or molecules, and they can, so especially in teeth tartar or teeth enamel, they can be traced for, or they can be extracted from thousands of years old teeth. So this is one of the main kind of sources of evidence for milk consumption among humans. And it is through mass spectrometry pretty, so I mean, it's not easy, but these proteins are pretty distinct. So you can't even, in most cases, my colleagues can say, well, this is from sheep, this is from bovine, this is from horse, and so on. And in archaeogenetics, as in many other fields, archaeogenetics has basically kind of reconfirmed older archaeological theories about domestication. So kind of this, it really settled that point that sheep, goats, and bovines were first domesticated in the northern Levant around 7,000 to 8,000, 9,000 years ago. But then from there, it's really difficult, and still there's not a lot of data how it's actually spread across. So I mean, one way Eurasia and the other side Africa, and I think mostly for like technical reasons of preservation, my colleagues are much more, or archaeogenetics generally is much more interested in cold and dry climates than in hot and humid ones. So Africa is still a pretty, there's not a lot of work in Africa or say South Asia or Southeast Asia, because it's such because the preservation is just much better in a climate like Mongolia's. So so about 4,000 years later, my colleagues find the first evidence of dairy consumption among humans in Mongolia. But in between, there is not a lot of data. So they had this initial idea: what if we look at the microbes and compare them to present-day microbes? Could we, through phylogeny, through genetic inheritance, find or kind of redraw the migration route of these animals and the humans, and of course also kind of connected to those questions, all these old questions. Is it migration? Is it diffusion? Are there maybe several domestication events in different parts of the world, and so on? So this is kind of their early interest, and then and because they were really, so this whole strategy relies on present-day microbes, and they realized that there is very little published data on microbes, and especially on kind of non-industrially used dairying microbes. They were looking for social anthropologists who could do this work and do like in a way, kind of ethnography, but also collect milk samples on the way for them. And kind of through getting into this field of ancient DNA, I was really struck by, or there's this huge discussion, and I was struck by the lack of a theory of history in the whole field. So I started to work at an institute called the Max Planck Institute for the Science of Human History, where not a single historian was working. I mean, not just that they don't have a department of history, they just don't have historians. They have collaborators, and of course, and then you might be aware of the fact that ancient DNA has captivated the science journalism over the last decade. And of course, Science and Nature and Cell they basically run the big ancient DNA study every 4 to 6 weeks. So it's a huge field, and huge progress has been made, and kind of the number of ancient genomes has grown exponentially over the last 10 years. But there is very little in the form of theory production, or of course there's always theory production going on, but there is very little explicit theoretizations of what a genetic history actually is. And of course, historians have always made, or for a long time, made the point that history is not out there, that you can kind of reveal the history. And now ancient DNA is coming back and kind of retelling these easy stories of history that is just, and in a way, kind of genetic methods being a form of time machine that could actually kind of bring you back to a lost history, and a form of and that would usher in a new form of scientifically based history that would do away with decades of speculation in archaeology and history, because now finally we have hard facts on which to base our understandings of history. And this has often been done, and often kind of the narratives that are undergirding this idea are very old school ideas of migration, warfare, and of course it is, I guess, genetic history a form of, so it is a history that is interested in genetic relations, which of course is, as we all know, one form of kinship, but and especially when we look at historic or ancient populations, probably not at all. I mean, definitely not the most important form of relationality between individuals and between generations. And then kind of to make it even more complicated, or maybe do it in another way, I then kind of started to work with Tina Warriner, who has had for a long time this interest in the archaeology of microbes. And I first suspected that this was also a way of getting out of this heated discussion of ancient human population genetics. But then kind of starting to read about the stuff that microbiome studies does, I realized that there is another extremely problematic discussion happening there. And that is how I get to Hadza microbiomes and this whole idea of getting to a human baseline in microbiome studies through studying contemporary hunter-gatherer populations. And I would just think that, again, I kind of get to a very simplified understanding of history. And so I've just found Hannah Landeker's idea of a biology of history extremely helpful for disentangling these different threads and coming up with what I hope is a more complex understanding of living with microbes and other non-humans. Like, you have a question?
Frederic, you've got your hand up.
Yeah, thanks, Matthäus. Very interesting article. I was wondering if the notion of cryopolitics developed by Emma Koval and Joanna Radin would be helpful for your work, because on one side there is the question of access to the cold chain when you have to bring frozen samples from Mongolia. And at the other, on the other side, I was wondering if ancient DNA could be practiced on naturally frozen samples, such as permafrost or things like that. And I myself used the notion of cryopolitics to think about the current controversies on COVID-19, where there's a lot of discussion about frozen samples in markets and how to freeze viral strains in collections in labs. So is how do you think about the question of freezing in this work?
Yeah, that's an extremely good point, and I've actually kind of only started to read that work a bit too late for including it in there. But I think this is extremely helpful to think of ancient DNA, especially as you said. So I mean, first of all, of course, we had to we came in with possibilities of freezing samples, and it was most interesting in our, we did like a 10-day horse riding trip to meet some of the I mean, to meet the only group of people in northern Mongolia that practice reindeer dairying, and so we had this huge, this huge container that you fill with liquid nitrogen, and then it holds I don't know, minus 100 degrees for 10 days, which actually developed for bull semen. So kind of another extremely important form of cryopolitics for artificial insemination in industrial livestock breeding. But as you mentioned, the other thing that is, of course, very very high on the list of priorities for ancient DNA research is actually permafrost. And this is the main source for really old and well-preserved DNA. So of course, there's been, for example, Beth Shapiro at Santa Cruz, who has been working on mammoth genomes, and of course the mammoth is kind of the most characteristic of these kind of extinct megafauna that people are actually trying to bring back into life. So Siberia is the, and it's interesting. I think I haven't seen anything from the North American permafrost, but Siberia seems to be a place where every few months there is a new find of really old, an old corpse of dogs, wolves, mammoths that are. So, and this is also how ancient DNA has been able to kind of to date back their oldest sample. I think now it's like 700,000 years that they have been able to extract DNA from. So yeah, this is an extremely interesting way of thinking through these issues.
Ashley, go ahead with your microbial care question.
Hi, thank you, Matthew, for this wonderful article. I have a question that is kind of going a little bit backwards, maybe to the dairy products, but also in relation to to genetic relations between microbes and humans. I'm really curious, what you talk a lot about care in this paper. What you really think that the scientists care about, and it's kind of a question about the ontology of microbes. So, how do they imagine the microbes when they care? Is it a strain? Is it really a code that they see flickering on the screen they care about, or is it an imagination of the microbe, what the microbes can do for humans, or is it something entirely different? So when scientists care about microbes, what is this thing they care about?
Right. So yeah, I think it is both, a little bit of both of what you said, and I think it's also more. So metagenomic it's, as I understand it, is extremely complex and really extremely complex to understand what you're actually looking at. So the way how this works in most cases, and of course that's sort of kind of, I was talking about purification and amplification, but this is of specific strains, but this is kind of a very specific subset of questions, because in most of the work that my colleagues do, they do work with metagenomic assembly. So they get a sample, they clean it out from all the non-bacterial DNA, but then what's bacterial DNA is basically dissolved into this letter soup of base pairs, or not base pairs, but it's pretty short fragments. So ancient DNA is also characterized by short DNA fragments. It's also one of the main ways how they are able to filter out modern DNA contamination by kind of the damage pattern, as they call it. So we are normally talking about DNA pieces of 30 to 150, 200 base pairs. And then, so like once they identify all of those, they then reassemble them in the computer and on the dry lab side of their operation. And then what they actually care about is, so they still have, in a way, because they still have to rely on whatever imperfect species definitions they have, even though they know how problematic they are. But what they are more and more interested in is what they call biosynthetic gene clusters, that are kind of so specific kind of sections of the genome that are also a few genes that are often kind of in close proximity to each other, that might actually be able to tell them something about something that microbe was able to do. So in a way, again, to kind of how a microbe produces specific small substances, small molecules, is something they're getting more and more interested in the form of getting to the question of what they actually can do. And again, these biosynthetic gene clusters are in a way also practical as a transgression of the species concept, because they seem to be also highly mobile between different individuals and even across species borders. But what I find really interesting kind of in your question, to me, I feel like the way how they actually, as you said, kind of do they look at code that is flickering on their screen. This is, so I mean, some of them are, because of course it's such a diversified division of labor. Of course, there are like the bioinformatics people who write code and maybe also kind of and do like the initial analysis, but then the way how it is further processed and presented is, I think, very much a graphic design practice. So you often have these matrices where you have 4 or 5 or 6 different dimensions, and on 3 or 4 axes. So they do work really closely with graphic designers to actually turn all this complex information into plots that are readable, and that are readable by a wider audience of natural scientists, not just people who are like in the core field of microbiome studies. But I don't know if that's actually getting to the bottom of
well, that is really interesting. Thank you for this answer. And yeah, the graphic design part of this is super interesting. I'm also working with scientists who are working on the marine microbiome, and so I'm asking them these questions too. What is it you really care about when you look at marine microbiome? But it's a really tough question, and you answered it. Yeah, in terms of caring, in terms of interest, in terms of curiosity, right? Is there also an affective component of care in this? Well, metagenomics is very computational, right? It's very much about, so what is their imagination there? What in terms of not just, or would you collapse that the interest and curiosity is the same as that's what they care about? Or is it another dimension of affect that is somewhat different from from the specific interest?
I think for kind of individually, for some of my colleagues, there is definitely a more than that in their affective relation. So Tina, who is the kind of the head of the lab, is, I think, very committed to what I also describe in the paper, to preserving microbes, food microbes, and to, in a way, see them as a form of commons that has to be protected from commercialization, and also, in a way, an interest in good food, I think, is for many of them kind of an important reason for their work, to actually to protect these microbes, but also, of course, to understand better how they affect us and how we affect them.
Thank you.
Thanks.
Okay, you got another question?
You're muted.
Okay, when I first read your title, Preserving the Microbial Commons, I get excited because, yeah, I was also trying to articulate the microbiome, human microbiome, as part of the commons. So, but I didn't find so much things in your article about the commons discussion stuff, just at the end. So I would like to hear more. How do you frame them? I mean, do you see them as a common pool resource? How do you frame this microbes?
Yeah, I mean, you're totally right, and I'm in no sense an expert in the huge commons literature. I mean, maybe for me, as a kind of someone who grew up on a farm and whose livelihood has always been very much dependent on common pastures, that is kind of a very kind of practical understanding of commons, of spaces, and but of course also skills that can be accessed by a large part of a community, and I think that through some of the work that also the fermentation activists like Alexander Katz or David Asher have been doing, I think that's an important part of spreading the knowledge and kind of telling people that it's actually totally doable, that you can ferment in your kitchen, and that you can be part of that movement and kind of but make your own fermented products. That to me is an important part of a commons understanding of microbes, and kind of an ethics of sharing both the microbes and the practices that make them thrive, hopefully. So we've been also trying to do a number of workshops at academic conferences where we make bread or cheese and talk about fermentation and people's personal practices. And I found it always extremely interesting how easy it is from kind of making bread together to talking about a lot of affective relations that people have with fermented foods, and a lot of childhood memories that come up through these forms of commoning, I'd say.
I asked this question because I remember when we invited Lorimer, you commented that you don't believe that these microbes will going to extinct because they're all around. So I was just, when I was reading your piece, I remember your comment when you were making about the extinction of these microbes itself.
You know, that's a big question, and I've heard very different opinions about it from people who are much more qualified to answer that question. But I mean, I do believe more and more that it's much more, that the danger is much more that the practices, the human practices, get extinct than the microbes. Of course, there is the possibility that, and especially when it comes to lactic acid bacteria, who are actually very fragile when it comes to antibiotics, that we are in a process of maybe not extincting whole species, but extincting important strains and lines that maybe have been fermenting with humans for a long time. But generally, and I guess this is one of the biggest, how to call it. I mean, it's an irony that actually in all that antimicrobial resistance is such a strong thing, because I mean, if it wasn't, we would have probably already killed this whole planet by the mass application of antibiotics over the last 50 years. So I think we don't have to be too concerned for the microbes. So it is in a way kind of, it is a very human-centric fear that I have, that if we lose the practices and if we lose the cultures of cultures, then it doesn't make too much sense that, or I mean, it's great that the microbes will still be there, but if we have no relation with them anymore, then this whole field of relations will be lost.
So I've got a couple of comments and then question, and also I'm going to have to leave about 10 minutes early, so Rachel's going to take the reins after I have to go. So first, in terms of comments, well, you know, one thing I was thinking about is in relation to how you might theorize the commons. So I just put a classic article by Anne-Marie Moll and John Law in the chat, thinking about regions, networks, and fluids. And you know, there's a certain topos to the territory of the agrarian commons, and I'm wondering if a network topology might be a way to theorize the commons moving forward in this relational way. Another comment just on ancient DNA, and this comes via Simon Park, a microbiologist who's also dabbles in art. He shared a paper with me once about the microbiological communities living on thawing mammoth tissue, and looked at the ways that ancient DNA was actively taken up and repurposed by these contemporary microbes. So I guess, you know, time works really different in these microbial scales. So you know, maybe also thinking with Astrid a little bit here about these hauntings from the Pleistocene or from whenever, might be a way to think about how modes of being and living from a past might be reanimated in a present and a future. And then finally, the question, phage comes up a number of times in the paper. You talk in the footnote about these industrial dairying practices that rotate starter cultures every few weeks to get rid of phage. So maybe just sticking with that line of thought that you were developing around the preservation of practice, and maybe again thinking with Anne-Marie Moll about a praxeography of microbial culture. What are some alternatives to that rotating of the starter culture every few weeks that might promote phage diversity in a post-Pasteurian way that would enable these more convivial flourishings of milk cultures?
So yeah, I think doing it the old way would actually help a lot for both phage diversity, but also kind of not the kind of build up and kind of these knockouts when phages just destroy a whole production because they just because they attack the main strain that's in the industrial culture. So if that, and I guess so this is the main reason why these natural waste starter systems are so resilient, because even if there is a kind of a phage infection that would clear out one whole strains of phages. In most cases, phages are highly strain specific, so often they can't even attack kind of a whole species. So this is why these cultures are so stable, and they can be used over months and years without any change to the system. So once the system works, of course there's other reasons why they are in a way fragile, but phages is basically not a problem. But then that's of course that's an interesting question. Could you scale it up to an industrial form of dairying? I don't know, would be interesting to look who is kind of, what's the biggest kind of producer who does it this way. I know that until very recently in Switzerland, a lot of the kind of, yeah, small scale, but still relatively large, kind of small scale industrial local dairies did use natural waste starter cultures, also because for economic reasons, because they're very cheap to if you kind of, again, if the practice is there and if you have someone who has the skills, then it's pretty cheap to keep them. But of course, on the other hand, it's an interesting question how much of it would be possible now, because the other thing that normally does not get talked about when we talk about milk hygiene is that it's not just the pasteurization and the sterilization of the milk, but also that the antibiotic treatment of the cows has been an extremely important part of why milk is the way it is today. And so that's the other question. So one of the kind of my main interlocutor in that piece said he was, for a few years, he didn't go to he wasn't an alpine cheesemaker, but he had a small farm in the valley, and he would only milk by hand, and he would sell it to the local dairy, and always get like a fine for low milk quality from the cheesemaker. But then when he stopped when he stopped milking his cows because it was just too much effort for too little money, he then learned that the cheesemaker was complaining that he couldn't get this one guy's milk anymore, who he always kind of gave too little money because he said the milk is bad, because he always used that specific milk from my friend to make his culture, because it was more contaminated, it was more lively. So yeah, that's an interesting question, and I would actually that's an interesting question to move forward and to ask people in the industry if they have ideas in that way. And just one comment to your ancient DNA comment. I would love to take a look at that paper, but I just thought of one of the first kind of small proof of concept studies that Tina is doing in her new grant, a collaboration with a modern molecular biology lab, and that is that they find the same group of Treponema bacteria on bones in museum collections and on the Mongolian cheese samples that we have. So apparently there's also something strange and interesting happening with kind of bones who have been in storage for a long time by and our new cheese from the steppes.
Matt, I was going to ask you something, but I want to make sure that everybody else has time to ask their questions first, if there was anything else. I was wondering, one thing I was wondering was just if you could say a little bit more about the lost tastes or the lost flavors. And you've been circling around it a bit. And I just wonder if you could say a bit more about, when you're conducting interviews, is everyone sort of referring to the same, is it always a reference to the same thing, or are there different farmers kind of referencing different senses of loss, if that makes sense? Because it sounds to me like, just based on your recent comments and what I read from your paper, you did try to kind of talk about, sense of nostalgia. That's something you addressed in the paper, but you just mentioned just now about, say, driving down prices for people's labor, or use of industrial starters. So it seems like there's a number of things at play, and I just wonder what else has come up in interviews that may have captured people's sense of lost taste expressly. What is their own sense of what has happened, and is it specific to a particular moment or event, or
Yeah, I think that's one thing that I still feel. So this was one of my kind of main questions that still feel not very well answered. And I mean, this is one of the things that I really can't blame on the pandemic, because I was really trying to do interviews with old cheesemakers, people in their 70s, 80s, and that of course was just impossible over the last 12, 14 months. So I definitely, once everybody is vaccinated around here, I really want to get back to that question, because I also, so this question of lost taste, but also the other thing that I'm really curious about is to know if one can still find memories of kind of a pre-Pasteur age. So how did people talk about microbes before, or how did people talk about cultures before microbes? But to the question of what was lost, I think there's of course a lot is happening, and a lot of people, and especially farming communities, have a lot of there's also a big discussion about a sense of, and this is a kind of a lost feeling of autonomy, of being able to live off the land, but also kind of, you know, live off your product, because nobody in the European Union lives from selling their product, because everybody now lives from subsidies. So this, of course, is, and I think it can often be kind of construed as a form of just like, you know, these whiny farmers. I mean, what do they complain about? They get paid for their services, but of course the whole system is really perverted when food is basically for free, and the food producers get paid tax money instead of kind of making sure that food is actually sold at a price that is appropriate to kind of the labor that goes in, and also in a way the quality that should come out of it, out of that labor. So that is definitely one thing. And as to that's interesting, kind of, if I don't think that so far I haven't heard anybody say this is the moment when it changed. I think kind of when the taste changed, but I think in a way it often is connected to specific people, and especially when it comes to like the summer pastures in the Alps, to specific Alps and people who worked in these places and who made a specific style of cheese. Which of course is also, over the last 50 years, there's been an extreme decrease in the variety of most agricultural products. I have a friend from northern Italy who said he has seen a map of the region of Trento from like 100 years ago, and I think there is like 150 or 200 grape varieties there, and now it's only 3 or 4. So this, of course, is happening in many sectors of agriculture, that we have this increasing uniformity in the recipes and in the products, and this is in a way also interesting for Rest, the main interlocutor in the Swiss Alps in my article, who is actually who decided to forego the certification of this Berner Alpkäse, kind of the local DAC, kind of the local protected brand, because he says it stifles all innovation, because everybody has to make the same cheese, and you can't experiment anymore. And he says it's boring if you always have to do the same thing, and if I can't try something new, without kind of introducing any industrial or genetically modified practices or something, but just trying to tinker with his cultures and continue with his care practices.
Thanks, Matt. Did anybody else have final questions or comments for Matt?
I just wanted to say that as I'm an archaeologist and I work in the Bronze Age in Europe, so this is all we're all kind of converges. But this has been a super interesting piece to think about, the piece that you wrote. merging ethnography with, the microbiome and ancient genetics and the plague and Yersinia pestis and all of those coming together, kind of moving around. But I understand that what you brought up, the third science revolution that's going on with the narratives, with the big human genomic projects in archaeology, and there are certainly lots of problems with them. But one thing that I kept thinking about was process archaeology, and I think that might be helpful to consider this, because it's taking off of North Whitehead and in a way, and they use pots and whatnot to use this, but it really for bacteria and for microbiomes, this is a really kind of beautiful way to maybe approach it. So that may be a lens to kind of look at the archaeological material and consider it. Just a comment, but this is excellent and really appreciated it.
Yeah, thanks so much. We also we've been tinkering, or my colleagues have been tinkering with methods of extracting dairy microbes from wooden, kind of more historic dairying tools. So I was able to sample a few like 19th century wooden milk containers in Switzerland. So I really hope that this is moving forward, and of course, that would be that would be a great kind of intermediary step from like the Bronze Age to now, to have like a kind of pre-industrial timestamp of microbial communities. But of course, yeah, there's also lots of work going on the archaeology, kind of on old pots, and that is definitely something that's extremely important.
Okay, well, thanks everybody, and good to see you. I was just going to add that we're going to do a double header this week. We're going to meet with some Notre Dame students and their professor, Eileen Botting, tomorrow, actually, to talk about their pandemic memoirs. So we're actually going to do 2 events this week. And then next week, Reading Group's going to meet with Klaus Herschel to talk about antimicrobial resistance. So we'll see some continuation with some of the concerns that Matt raised today as well. So good to see everybody. And Matt, thanks so much for joining us, and congratulations again to the new addition to your family, and just
Thanks,
wonderful
everyone.
new piece.
Really great.
We'll look forward to seeing this piece coming out in Cultures of Fermentation in the fall. So very long-awaited special issue.
Thanks, everyone. Have a great week.
Thank you.
Thank you. It was amazing.